Okay, so again, thank you very much. I appreciate it, Shelby. All right, again, super formal. We're going to zoom through some slides because you've heard a lot about the HRDP already. My name is Laura Saba. I'm out of the University of Colorado. The original intent of this workshop was to talk a little bit about the transcriptomics data that we have and how to access it now through our REST API and our R package. Try saying that several times fast. But being that this is the Complex Trait Consortium meeting and it is a little less formal and we do appreciate a lot of discussions, I may throw in a few really opinionated things, hoping to provoke some discussions around some of these things. So this introduction to the hybrid, first of all, I do have to give credit to Hao Chen once again. Everybody has to give credit to Hao Chen, but many of these slides were originally from many of these slides were originally from a webinar we did a couple years ago that talked about the HRDP. So I did steal a lot, and a lot of it is his words rather than mine. But we'll talk a little bit about the rat diversity panel, try to hit on some points that may not have gotten hit on earlier, but skip through a lot of points that did. And then I'll talk about Phenogen's transcriptome resources. So for students or maybe people that haven't been formally introduced to this idea of inbreeding, there's two religions in this world of genetics, and Abe belongs to one and I belong to the other. And so we're diametrically opposed. No, I'm just kidding. So Abe and Leah have this beautiful HS resource, which is heterogeneous animals that they're using for testing. I came from the school of inbred animals. So started off working with the BXD, realized there's a lot of things we can do in rats that we can't do in mice. And a lot of the RI panels in rats are significantly smaller than the BXD's 100-some strains. And so, again, like I said, I'm going to jump over here. So what we do have in rat are these two recombinant inbred panels that are around 30, and then we have this multitude of inbred strains. So I won't go into the history of these two RI panels, but by putting them together, we increase our N. So now just instead of having 30 strains, by creating these hybrid diversity panels, then we get to 100 strains. And one of the things for a statistician that makes me real excited is that by adding these RI panels and these inbred strains, we get that boost in power because we have those two RI panels, remember? So we don't have for many of our loci, we have very high minor allele frequencies, but by throwing in those inbred strains on top of those two RI panels, what we can do is break down some of those haplotype blocks. And as Abe beautifully introduced earlier, there are some benefits to thinking about a renewable resource, so thinking about testing animals when we know what their behavior would be if we tested them in another paradigm. So we can do that in a little bit different way than the inbred, so we do miss on some of the benefits that Abe talked about, but we do gain in other areas. And I make the joke about two religions, but it isn't a competition. And Abe and I have always talked about some interesting studies that we can do using the HS and the inbred panels as almost a discovery and validation cohort. In fact, we were lucky enough to get some money in collaboration with a group in Boulder to do just such a thing, and looking at oxycodone consumption and thinking about the data that Olivier George is already gathering on the same paradigm, and how can we put those together both in the context of can we validate peaks that they've found, but also for these behaviors that you're measuring so many dimensions, can we reconstruct some of these latent variables in the same way in totally different populations? So just like they're doing in human GWAS studies, where you likely can't get it published if you're just doing a GWAS in a single population, but thinking about doing it in more. What we have the advantage of, again, and why I like it, and that again is my geeky statistician coming out, is with these inbred panels, we can start layering all that information on top of one another. So we can take the proteomics and the metabolomics and the RNA sequencing from these strains and the genotype information and layer them on one another. And so it really highlights some of the work that RGD is doing to bring those data sets together. That's really going to be powerful as we move forward. And the same thing with Gene Network. We talked about the hybrid rat diversity panel, and I did add I know, I did add the reference. I'm not throwing any shade here, but yeah, sorry. So I am going through this really quick because we have covered all of this and I want to be respectful of your time. But obviously RGD is a huge resource when it comes to the HRDP and live rats and rat tissues coming from Mindy's project, and then all of that phenominer stuff that you heard earlier and all the great things that they're doing. How has done a lot of genome sequencing information on these strains and has that bioRxiv paper that was mentioned earlier that's currently under review. And then I want to show you some of the deep dive that we did in transcriptome data just for the HRDP. And then finally, there's lots of really fun tools on Gene Network. And so I'm going to skip through all these. Sorry, guys. I know you want. I feel the pressure of a 4:30 talk on a Monday. I'm feeling it. So you've heard about all.
Everything's slow here.
Well, if I was in Colorado, people would be like, it's time for my hike. The sun's going to go down. You got to let me out. So what is Phenogen's contribution? So if you haven't heard of the Phenogen project, we've been around for a really long time. I actually started working with Boris and Paula in Colorado when I was a wee student, and they were still spotting their own arrays. So even before Affymetrix, RNA-seq, those two were spotting their own arrays and thinking about the power of measuring RNA expression really deep in some of these large genetic panels. But here is where I'll stop skipping and go real in depth. So what we have as far as HRDP RNA sequencing is we have two sets of RNA. So whenever we extract RNA from a tissue, what we do in this project is we separate them out by size. So we take the group that's long, so over 200 nucleotides, and put that into a little bin and make a library out of that, and take those that are less than 200 in size and make a slurry of that and do sequencing on that. And so why we've chosen to do this total RNA as opposed to pulling some of the only the transcripts with the poly-A tail. So a lot of times with your standard RNA sequencing, we pull everything with a poly-A tail and sequence those under the guise that most things that have a poly-A tail are protein coding, and a lot of the things that don't have poly-A tails are non-coding, and that's a generalization. Obviously, there's some that go either way that have poly-A tails and are non-coding, and some that are non-coding that have poly-A tails. But our goal was to really characterize all of the RNA in the cell. And so we took that total RNA that's really long. We did a ribosomal RNA cleaning, just meaning if we weren't going to filter out some of the ribosomal RNA, that's all we would be sequencing. We wouldn't actually get into anything else. So we do a first pass, a couple pass on some tissues, a third pass on some other tissues to get rid of as much of that rRNA as possible. So where our current databases are, and they're on Phenogen website, is our current version 6 of the HRDP has 58 strains of the 96 strains in the HRDP. So we're doing strains as Mindy rederives them. We go really in depth to individual samples. So we're looking at like 60 million paired end reads per sample. And then we always collect multiple biological replicates per strain. So we have a really good idea of the between strain and the within strain variants for all of our genes. We have some sex data, but our main two databases are this brain and liver for this entire panel. And then we have a little bit of data on heart and a little bit of data on kidney. We version control everything. So we're on version 6 right now. We're in the process of processing all this data for what we're calling version 7. So at version 7, we'll be up to 73 of the 96 strains, again, keeping our biological replicates high. We did run out of funding, so our 24 expired. So we prioritized the brain over the liver at this point until we get some more money to finish that out. We do have a Phenogen website. I'm not going to go over all the details of the Phenogen website. I'm actually going to focus in on this, what we call our data download area. Because all of you all are quantitative geneticists, a lot of you are bioinformaticians or biostatisticians. And so what I'm hoping is that you'd find these big data sets useful and you'd like gobs of data rather than focusing on a single gene at a time. And so I want to tell you a little bit about how we process this underlying data set, so for you to download. And so if you go to different versions of our RNA database, so I just looked up all of the information on that we have on brain RNA on version 6. And so I wanted to take a minute to just justify myself here a bit. So thinking about some of the things that Abe alluded to too, we can analyze and do our RNA-seq either at what we call the gene level, or we can do it at the transcript or isoform level whenever we're quantitating. And so there's advantages and disadvantages to both, right? So for isoform and transcript, we really understand a lot about what oftentimes these different transcripts have really different roles in the cell, either because of where they're being distributed to or the proteins that we produce. So oftentimes it's really, really important to get down to the isoform level information. However, short-read RNA-seq, what we're doing is aligning short reads back to the transcriptome. So sometimes it gets a little bit hard to quantify the individual transcripts. So there is some uncertainty that comes into annotating these transcripts, annotating these isoforms. The other drawback is if what you're doing is going to look at your KEGG database, you're looking at gene ontology, those are all annotated at the gene level. So doing individual isoforms isn't going to get you into those enrichment results if your top priority is to do find a biological pathway that's important. So hence us giving you a ton of information. So what we do is we actually have 4 factors that contribute to our different data sets. So the first time we use either our ensemble for version 6, so that's the version that's up right now, or in our coming version of version 7, we have switched to RefSeq. And so for those of you that are connoisseurs and care why we switched from ensemble to RefSeq, we did do a deep dive into the 2 new transcriptomes for RN7, and RefSeq had many more individual splice variants annotated than ensemble did. Ensemble lost a few categories, so it's got really low number of microRNAs that are annotated anymore in ensemble when there were a lot more that were. There's still some discrepancy between the 2 annotated databases, but RefSeq does tend to be bigger in this case, where for years and years and years it was smaller, and ensemble would have been thought of as the more comprehensive transcriptome database for the rat. So we have either that annotation level, or we do a reconstruction where we, and I will go through that in more detail, where we say, oh, we admit we don't know all the transcripts that are being expressed in brain. We're going to let the data tell us a little bit more about some different splice variants or transcripts with different polyadenylation sites that are being expressed in our data set. We can do that different levels of quantitation, of either at the isoform level or at the gene. And then we also report data out to you both at the individual rat level and at the strain mean level. we've realized that not everybody loves data as much as I do. And so sometimes it's easier to get 73 strains than it is to get 210 samples. So that's why we have so many data sets in this file. But when you want to download our data set, you have some choices and want to think about what you get there. So why this transcriptome reconstruction? I have just been in awe of the transcription machine that we have. Whenever we look for for differences in splicing, we're guaranteed to find them. But one of the things about rat, and actually this graphic is much more impressive now with or much less dramatic, more hopeful, with the switch from RN6 to RN7. So if you can see, what I'm doing here is I'm comparing the number of genes across human, mouse, and rat. Human, mouse, and rat have about the same amount of genes. We've always found about the same amount of genes. So it's not like we're missing totally new novel genes, but when we start looking at things like splice variants that are annotated, you can see there my big difference between Ensemble and RefSeq. Ensemble has less than 2 isoforms per gene, which we know is wrong. RefSeq is much closer to what we're seeing for mouse, but human is still in the lead. And again, I don't have necessarily the scientific data set to say that this is definitely not biological and more is a loss of annotation, but it is my opinion. I told you you guys would get a few of my opinions as we go through this. So what we do on that.
Or it's only thing that makes humans special anymore.
Is opinions or splice variants? I'm kind of losing. So what we do is we use one of the classic transcriptome reconstruction methods of string tie. So there are a few out there. String tie is kind of the most popular, and string tie tends to be very, very good at finding exon junctions. Right? The challenge to this research, reconstruction, is putting the right exons together. Finding those exon junctions isn't terribly hard, but putting it all and chaining it together is what makes it a challenge. And string tie has notoriously been terrible at finding the ends of transcripts because their goal is to be as inclusive to as many reads as possible. So their tendency on that algorithm is to push those transcription start and transcription sites as far out as they need to incorporate a lot of reads. Where we know that there are some important differences in polyadenylation, and we know that there's some differences in polyadenylation within the same tissue that are important to where that transcript gets shuttled to in the cells. So we've added on top of that string tie a new machine learning algorithm that we use called HRDP, which incorporates some of those canonical sites for polyadenylation along with the read buildups from the RNA-seq. So it's taking both from the DNA and RNA so that we can define those ends a little bit better. And so it's a very iterative process for us. We do it in multiple steps. This is just an example of one where we published with just using the HXB that you can see in our iterative steps. Once we get down to transcripts that are detected above background, we end up splitting our samples pretty well against those that were identified between string tie, those that were modified from string tie with HRDP, and those that were in the initial Ensemble database. So we are confident that we are getting more information about individual splice variants. And so we've done that for all our data sets. So it takes quite a bit of time. I meant to put a picture up of my hero, not just Hao Chen, he's one of them, but also Spencer Mahaffey, who's a computer scientist in our group. So with these large data sets and running this iterative process, he does make use of some supercomputers at the University of Nevada in Las Vegas to be able to push off some of this data, get it to run in process, turn it around and run it again. But in the end, that's why we have different versions, and that's why version 7 isn't out quite yet. We're waiting on one data set to run and run our final iteration for that detection above background. If you go to our website right now, we have this data download page, and you could literally go down and click the download data and get those individual data sets that you want off of the website. There's lots of tables, there's clicking here and clicking there, but we recently became, lots of clicking. We recently became, not recently, let's be honest, it's been a few years. We became part of the Consortium on Addiction Data Integration, so CADI, which it was an effort led by Alyssa Chesler. And this is where my opinion in my soapbox is going to get up there. So the opinion of the consortium was that we've got lots of interesting websites doing a lot of different things. And it can be extremely overwhelming for new users. It can be extremely overwhelming or frustrating to have this data set here and that data set here. And when can't we get some of these tools who are actually, most of these tools are very unique in and of themselves, but is there a way we can get tools communicating and data sets communicating? And so part of that process was to agree to a REST API structure and think about moving all of our data to be accessible from REST API. So Phenogen has started in on that process. And so these are kind of the first steps to it. So those high-level users, so for people like RGD, if you want access to the latest and greatest version, we have a more sophisticated Phenogen REST API function that I'm told spits out JSON stuff. I don't know what JSON is. I tried to look at it and got completely intimidated, but I'm told that people like Piotr would think it's wonderful.
Don't count on that.
The concept, he's not going to say my stuff's beautiful, but the concept of putting something shareable in that way. So in that vein, to make people like me happy and not just the Piotrs and the Spencers of the world, I convinced Spencer to also make it part of our package. So now he's developed an R package with the REST API. So if you're a big R fan like I am, you can get access directly to those data sets through R. And so to just take you through it briefly, it's still in development, so you do have to install that dev tools and install it from the GitHub repository, because we're working on it, trying to make it shiny, not shiny because that means something in R, make it pretty and usable and all of those things. But the idea here is that he's created a set of functions that this first function just tells you what all the data sets are in our database so that then you can call the data set that you want. And because this hierarchy that gets so confusing with the genes and the transcripts and ensemble versus the reconstruction, we do add a few steps here to help you narrow it down and do a stepwise phase. So once you decide that I want the HRDP version 6 whole brain, then you can go in and say, okay, what data do you have for that particular set? Right, so here we're telling you I've got ensemble, I've got reconstruction, and that line 15, we probably need to come out with a better word for it, but it's actually the GTF file that defines the reconstruction. So it defines all of the genes and transcripts in that reconstruction model. Then we get once you have that, then you can say, okay, well, now that I've told you I want the ensemble files, how do we get down to the actual ensemble expression data? And so that's one more function to list all of those. And then finally, we can get to that data set. So here I've just showed you the first 6 strains and the first 5 transcripts from ensemble. So those first few steps you don't have to do every time. It's just a tool to help you find the data set that you want and be able to call it in a very smooth and efficient way. But once you've found it, all you have to do is this step where you pull that data set in, and then you automatically have it. If you've never used the BioMart package in R, you can pull in all of your annotation information into R then too. You don't have to download a bunch of files from different sources. You can pull it all into the R to annotate your data. And then just to demonstrate, if we want to look at OPRM1, we can see in our annotation file that there's 2 ensemble transcripts for OPRM1, and we can pull out that particular data from that brain database. So again, in a few simple steps, you've pulled out that data, you have it in hand. If you want to correlate it with a phenotype, if you want to run and develop some fantastical networking or co-expression tool, this is a great data set to do that with, because it is so deep. It has so many samples that you can use. Yeah.
I have a really naive question. What are the units of those numbers?
So yes, no, that is not a naive question. I did skip over that because I was super excited to show you this. So as you can imagine, this is like 200 samples, right? And we've been rederiving it, and as the rats get rederived. So we do these. I keep saying that. My children would be laughing hysterically. We have lots of batches that have happened over the year. So the way we process our data is, once we get that count data from our RSEM, is we do a regularized log transformation. And what that does is, it's basically a log to transform, but it adds a pseudo count in case you have 0 values, because I want those zeros to still be in there. So that first step, what that does is get my data into something that kind of looks normal and accounts for some of the differences in library size. But my R-log data still shows really large batch effects. So one of the tools, and we've explored a couple different ways to handle the batch effects. And what we've found, because how we've designed our batches, is that combat, and I'm not sure what it actually stands for, but it's called combat, and the C is capital and the B is capital. And I'm happy to share that with you at another point in time. That is a normal batch correction algorithm. And so that's the batch correction algorithm that we used on those transform data. So this is not your TPMs. I know how y'all like those TPMs, but we are on Geo. We're one of those super series that are super obnoxious. And we do have some count data up there. So we do have that other places, but we have found, for purposes of making it a universal quantitative measure, that we can use a bigger statistical toolbox to analyze than simply doing something like DE-seq on, that we use that R log followed by a batch correction. So these are, you can think of them as a log 2. So a 1 unit difference is a 1 unit fold change. But it's a good question. There's lots of opinions on how to analyze it.
Yes, my question is that it's wonderful for comparing the data within Phenotype, but how would our GDUs take in stuff from Geo? How would we normalize, how would we utilize this so that you can compare this super series with this super series?
Well, why would you want to compare our super series? It's all inclusive. I know. So yeah, that's a hard one, right? Because not even because we're using these R-logged values, but we're also using this batch correction. And that, it's hard to recreate that batch correction and get down to the literal expression of it. If I were Rob, I would tell you to do a Z transform and get it to be all on the same units. If I were Abe, I'd tell you to do a quantile normalization to a normal. Yes, but I am not Abe and I am not Rob, and I would prefer an R-log, yes. But I'm willing to duke it out over it.
Maybe the right way to think of all of this given that you have a superset of data, and somebody else has a superset of data, as just a mechanism to find data sets that are good to address the question that I have. And then if I could get to the underlying data that was used to derive numbers, I can bring on, on the myriad sites where the data exists, I could pull all of that together and give you what it is.
Yes. And so, yes, that is one of the answers to it, right? So what the purpose is, or what I interpreted the purposes of RGD's website and the TPMs, is to talk about global expression levels of genes, right? For that question, the answer is TPM, because that's a normalization to tell me how much is expressed in that tissue. For the R-logged values, the purposes of R-log values are to be able to compare among the strains within a gene, right? So the R-log values from gene to gene aren't necessarily interpretable, but the R-level values within a gene are more comparable, or we can start doing things like co-expression. So from a very technical standpoint, it depends on what your question is. Right? I'm a big advocate of R-log batch correction when what you want to do are things like co-expression, QTL, looking at differences between strains within a gene. If what you're wanting to do is say, is my gene expressed in brain, then you need to do a TPM. And to me, those are different questions, and they're both very, very useful, but they're different questions and we would require slightly different pre-processing. So they make fun of statisticians because our answer to everything is, it depends. It depends.
You want to look at the data compared to the HRDP in the brain for what you've done. How would I do that?
Qualitatively, I would do it with a TPM, but I would not be confident going from one experiment to another experiment and saying these are definitively differentially expressed, unless you had some way to account for batch, to account for differences in experiment, to account for those different things. But I do think you could qualitatively say it, right? I just wouldn't attach a p-value. But I'm also a little conservative with p-values. You can then see what your distribution obviously looks like across your strains. If you have a few strains that you're particularly interested in, obviously you could do the differential expression between two strains. Our reconstructed transcriptome, what we do to be able to define what those genes are, we give you a GTF file. Like I said, this is still a little rough. Our GTF file kind of imports into R a little bit strange. You do have to do some manipulation, but we're working on it. But you get a GTF file. So GTF files, if you don't, it's gene transfer format. And so those are what you use whenever you do quantitation. So in RSEM, you use a GTF. If you want to visualize on the UC Santa Cruz browser, you can upload a GTF and it'll show you these transcripts right along with all the annotated transcripts and tracks that they have. And I'm assuming that it may be a possibility in JBrowse to upload a GTF file. We also have a couple of genotype data sets available. So we had the HRDP originally, the only data that we had for genotype was an old marker set from the STAR consortium out of Europe that did Affymetrix arrays to get SNPs on them. And so back in RNA-6, that's what we were using for mapping and things like that. So we have some older versions that are based on the STAR consortium genotype arrays. For version 6, when we moved to RN 7.2, we graciously used the genotype data from my hero, Hao Chen, and my other heroes at RGD. We supplemented those two together because Hao has all the RIs that have, those RIs that have been tricky to resuscitate. So, but we had phenotype data on them from the the Czech Republic. And so we mix those two together to get the genotype set. So we do some cleaning that involves checking out we have so many SNPs that we're able to let go ones that are missing genotypes. We're able to let go of ones that are heterozygous, and we have the privilege, the opportunity to be a little selective of what SNPs we allow into our mapping data set. And so we also do some looking for things like I was taught by Rob Williams to do, of looking for some double recombinants and things like that don't follow what we know the recombination should be, right? We shouldn't have things recombining back and forth within a small physical window. So we do all of that cleaning on it to get to this new data set. And that's a whole other bar conversation about some weird things going on in some of the RIs in a couple of regions that it looks like they have 3 founder haplotypes. But yes, I'd love to discuss that. I'm looking at you, Hal, to figure out what we can find out more about those. And so those strange regions, those regions that aren't following what we know at this point in time, we just took them out. And so we left those as blank regions and took those markers out with the fact that we'd rather have false negatives than false positives at this point when mapping, and hoping that the genes around, or the markers around that, would catch that signal for us. And those are just demos. So that's really the coverage of the talk. There's lots of this and other data sets are on Gene Network. Don't want you to forget about them too. We do, again, a little embarrassed to say, we do need to get version 6 of our HRDP to you. We have version 5 up right now, but again, to me, that's a case study on why we need to get these REST APIs talking to one another. So when we have that new data set and we release it, it can automatically be populated in Gene Network. It can automatically be populated in RGD. We can automatically send those, and it's not, oh, well, we'll gotta find a Dropbox or an FTP site to get those to you when somebody's paying attention, right? Is she sharing here? I think he already left, but his proteomics data is up there. And then phenotype, I'm gonna super skip over all of that because you all did such a great job of telling us with all the phenotype data. I did not even know that was all there. So in summary, we think it's a useful tool. I actually forgot to update those, so I'm gonna skip that. Where do we go from here? I know that Peter's doing a lot of single molecule RNA sequencing. We've done a few samples, and so we've done in version 7 we did do some playing around with string tie and being able to add in that long read sequencing as part of that string tie process. So that's an option to put it in there to help guide that reconstruction process. And we did do that. In my opinion, there weren't strong results for or against it. I think one of my most disappointing realizations in life is that we still don't. There's lots of those. I don't feel like we have a true gold standard in identifying all of the isoforms that are expressed in a tissue, in a cell. I do think there's lots of noise. I feel like every time we look, we find more. So I don't necessarily think it's the fault of the technology, but I do think the transcription process can be a little bit wonky and error prone within the cell. And so really figuring out which one of those transcripts or isoforms are biologically relevant is more of the challenge. And that's where we're moving to, not only with this detection above background, but one of the things that is unique about the HRDP is we can start thinking about which of these transcripts are heritable. And is that a sign of biological relevance, right? But we're getting more of it. We should be able to integrate it. we can use it. I don't think we can use only it or only the short read to really capture the transcriptome. We got to mix the two. There's lots of cool other omics coming out, and how do we mix? We have a whole grant application on how we mix single cell RNA-seq with this massive database we have on bulk to see if we can't disentangle some cell type specific expression from our full panel instead of doing 96 on single cells, but be able to do a few samples on single cell and tease out that other part and looking at those drug-bite interactions. A couple of other babies in here, but my baby at the very end is that differential co-expression. So really thinking about what disrupts some of these co-expression networks, what changes those networks when we expose them to drugs or other environmental toxins, and what's going on there. So yeah, I think speaking of bringing up conversations, I obviously plug the REST API and talking between sites and thinking about how we collaborate and not compete, and both with websites and data and populations and things like that to strengthen the whole rat community. Because we did talk at the very beginning that we do have some naysayers out there when it comes to model organisms. So I was hoping to get some opinionated people to really step in and take over for me so I can go have a beer. Or 20% ethanol, something 20%. Obviously, this was funded by a lot of different resources, and a lot of people helped with this. It's been a community effort. These are huge databases. And finally, my other very shameless plug is that for some of you know that we used to do a webinar series with our P30 grant that no longer is funded, but we felt like the web series was useful. So I'm twisting arms to try to get it restarted. So if you have topics that you're super excited to hear about, please let me know, and let me know that I will reciprocate by asking for a letter of support for funding eventually.
Thanks for making the plug for the REST API as a glue between different databases and analysis platforms. I just want to make just a small comment that it's not just between the websites, but also a glue for individual users, because they can now use all of these resources and do their own thing, create something new, and maybe push back to one of the websites if they like it. So I'm really excited about that, and we hope to work with these APIs and build our own tools to make different things interoperable.
Because Gene Network's moving to it too, right? Or you
Yes, Gene Network API we have now 3 APIs in 3 languages. Julia, R, and Python. We'll write one for Phenogen. We've written one for metabolomics workbench. So, and all of your things are we have it in the pipeline.
Where it's interesting, can you go back to your future directions or directions period? Yeah, that one.
Yeah, I copied and pasted that for the webinar, so it's definitely a duo.
Okay.
On the future direction.
So Laura and I and many of us are used to being nagged. Why aren't you doing everything single cell, you fool? I mean, bulk liver. Come on, there are at least 2 cell types in liver. Brain, there are at least 5 cell types in brain.
Some of us.
So I think we need to have a position paper where we say why bulk analysis rocks for if you're interested in the organ as a system, and if you're doing genetics and you know damn well that single cell RNA-seq is a cluster something or other when it comes to high noise. It's just awful. So you know, people have been talking about doing single cell genetics. Have you?
There's
Say-
a.
Yeah.
Yes.
Only a fool would do this. I tried it myself. So I mean, it's really hard. And it's basically, the question is why? It's really expensive, but it's getting cheaper. So the thing, if people nag you, and they've nagged all of us in reviews, you know, my counter nag is, if you're interested in the nervous system, you don't care about the level of expression of an isoform in the cell. What the hell does the cell mean? Did you mean the distal dendrite? Did you mean the axon? Did you mean a speckle right next to the nucleus? It's kind of a ridiculous question when you have neurons that are this long. And I mean, there are solutions to this, obviously, when you get single molecule reads. But okay, Rupert, you continue the rant.
Oh, can I turn on, is there a way to turn on those ceiling mics? There we go. All right, I turned on the ceiling mic. So it should be.
Rob, you can continue. But when you're talking about brain, so just to continue that thought, when you're talking about brain, a lot of people are doing single nuclei, right? If you're going to get single cells, complex cells from the brain, you can't sort whole dendrites, right? So your single cell is probably going to be single nuclei. So you're actually missing stuff. So I mean, that's a compelling argument for doing whole tissue.
I mean, I would soften the argument just a little bit to say that
Good
that
too.
that there are some uses for single cells, single nuclei, but I think the uses for them are about how we integrate them with other data. Because I do believe that in our bulk, we can start seeing signaling between cell types and signaling between brain regions that we would completely miss if we were doing a single cell. But the single cell information gives us a little bit more detail about what cell types may be involved.
I think it is good to do both. It does this ceiling like I think it is good to do both, for sure. I was impressed. There was a project I was involved with, actually Francesca Telese was the leader of the project. And they did single cell RNA-seq, and they also did ATAC-seq. And the single cell RNA-seq, I think, was interesting. But what I'm going to talk about is the ATAC-seq, because it turned out that one cell type, excitatory neurons, went completely in one direction, inhibitory neurons went completely in the other directions. Had you bulked them together, you probably would have seen no signal. But in fact, there was this incredible signal where it was diametrically opposed depending on cell type. And of course, we all believe that the state of the chromatin is going to have implications for gene expression. So there's also a similar pattern that you get there. So I think we're going to have to do some of both. Yeah. But single cell is really expensive, and I think it's like the easy thing for a reviewer to say, oh, just do it in single cell, right? It's like, it takes about 5 seconds to express that thought. And it's like you have to build in arguments about why you're going to do it the way you're going to do it.
Defend single cell, but it's right to point out that we've used Francesca's data. It was critical for us.
Yeah, I just think the argument is it doesn't completely replace bulk or nullify things that we've found in bulk. I mean, we always we've been hammered for whole brain for years. And our argument's the same as yours. Tell me what's the most important and only brain region that's involved in some of these addiction-related traits. Every person you ask would give you a different brain region. And so how do we do kind of that first pass to narrow it down so we can think about building a better functional hypothesis than doing a multi-million-dollar study on it, itty bitty brain region and then be off on the brain region?
Well, and then the whole thing is that you're not looking at cells, you're looking at nuclei. So you're missing a whole bunch of stuff that way, right?
Mm-hmm. And even single nuclei RNA-seq, there are people that are experts at doing it. That's not the standard core that can pull it off in a very precise manner.
They're experts, and they're experts who know to do flow sort. So Josh Sains, who developed this method at Harvard, was trying to do glaucomatous retinal models, and he spent 4 years beating his head against various single cell methods. And then he just did a flow sort and did single cell on flow sort, which I think is ironic. So even the best people struggle this, especially if you're dealing with old, cranky, half demented tissue from glaucoma or something like that, or Alzheimer's. It's progressively harder as you get to older cells. It's easy if you're dealing with neonates or immune cells are relatively easy because you can flow sort those reasonably well without damaging the cells.
And us bioinformaticians and biostatisticians need to catch up and be able to deconvolute some of these expressions too. Again, I think it's a integration of the two.
Are people burned out?
It is 5:20.
See that impression, Rob?
It's 4:20 in Colorado. I didn't mean anything by that. Okay, thank you guys for coming. Thank you, Rob, for a great.
I just wanted to find out one thing.